Myself, Mr. H.T. Mate, Department of Botany, RBNB College, Sri Rampur. Today we are going to study T.Y.B.Sc Botany, semester 2nd, and from the second semester paper first BO 121, Plant Life and Utilization 2nd. And from this, credit number 2, topic number 1, that is the gymnosperm. So today we are going to study the internal morphology of root and stem. So first, internal morphology of the root. In the cycas, there are the two different types of the roots are getting developed. One, that is the normal taproot system, and another, that is the negatively geotropic root. That root is called as the coralloid root. So this is the internal morphology of the coralloid root. Now, from external side, there is the periderm. It is a multi-layered. Inside this, there is the cortex, and cortex is get divided into the into the three layers. That is outer cortex. Outer cortex, here cells are loosely arranged. It is a multi-layered, and cells are somewhat thin layered. They are parenchymatous and they store the food material. Now, in between this outer cortex and inner cortex, there is another layer, and that layer is called as the algal zone. In that algal zone, cells are somewhat elliptic in shape. They are loosely arranged. And in each cell, there are some BGA, that is the blue-green algae are get developed. For example, Anabaena nostoc, some bacteria are also get developed. And these algae or bacteria, they are important for the atmospheric nitrogen fixation, which is symbiotic association in between this root of a cycas and this microorganism, that is algae and bacteria. So inside this there is the inner cortex. The most portion is occupied of the stem of the root is occupied by the inner cortex. It is a multi-layered. Here again cells are loosely arranged and they are parenchymatous and they store the food material. Now this inner cortex layer shows some tannin cells. That means there is a deposition of the tannin. Inside this there is the endodermis. Endodermis, it is single layered and it is the innermost layer of the cortex. Inside this there is a pericycle. Pericycle is a 2 to 3 layered in this cycas coralloid root. Inside this there is the vascular bundle, and here this vascular bundle is a triarch. That means there are the three arms of the xylem are seen. Now, in this xylem, in yellow color, this is the xylem. Here the protoxylem is toward the periphery. So, the xylem is the exarch. And at the center, there is the metaxylem. So, this is the metaxylem. The cells of the metaxylem are large. And cells of the protoxylem, they are small in size as compared to the metaxylem. And whole of this xylem is completely encircled by the phloem. Now, this is complete internal morphology of the cycas coralloid root. And there is a speciality of a development of the BGA zone, that is the algal and bacterial zone, which is important for the nitrogen fixation. Now, there is another root, that is the taproot system. Now, this is the internal morphology. These are the all cellular details of the internal morphology of the normal taproot system. Now, the external layer is covered by the cuticle. So this is the cuticle and this is the epiblema. The outermost layer, which is a single layer, is called as the epiblema, and externally it is covered by a thin layer of the cuticle. Now this is the xeropathic character of this cycas root. Above this there are the root hairs. They are unicellular. Now inside this there is the cortex, and very large part of this root system is occupied by the cortex region. Now again this cortex is get covered by, it is get divided into the three parts. That is the outer cortex. Here cells are polygonal in shape. They are compactly arranged. Now inside this there is a middle cortex, which is parenchymatous in the nature. Cells are thin or thick walled. They are loosely arranged. They are parenchymatous in the nature, and so they store the food material. Now inside this there is inner layer of the cortex. It is less as compared to the middle cortex. Middle cortex is very large as compared to the inner cortex. Now here, inner cortex cells are also parenchymatous. They are also thin-walled and they also store the food material. But in between these cells, there is a deposition of the tannin and mucilage is also seen. Now this is about the cortex, but inside this there is the stele. Now outermost layer, that is the endodermis, which is again the innermost layer of the cortex. Inside this there is a pericycle, and here also pericycle is a multi-layered. Now inside this there is the xylem and phloem. Now at the center in this yellow color there is the xylem. Now here again protoxylem is toward the periphery, and because of this xylem is called as the exarch, and at the center there is the metaxylem. Now in both protoxylem and metaxylem there are the tracheids, and other elements of the xylem are also present. Now this is the diarch. Sometimes diarch or tetrarch xylem is also get developed. That is xylem, diarch, tetrarch or triarch is also there. Now this diarch xylem is completely encircled, covered by the phloem, and in the phloem there are different types of the elements of the phloem. For example, sieve tubes are there, phloem parenchyma is also there. So this is about the normal taproot internal morphology. Now we are going to see the next point, that is the internal morphology of the stem. Now these are the two different figures of the two different age group of the cycas plant. So this is first, that is the young stem TS, and this is the old stem TS. This is the outline of the young stem TS, and this is the outline of the old stem TS, and these are the cellular details of the young and old stem TS respectively. Now first, that is the young stem TS, because there is a difference in the development of the xylem, in development of the secondary rings of the xylem, phloem and cambium. This is the major difference in between these two young and old stem. So first, that is the young stem TS. Now from outer side there is the periderm, and periderm is divided into the three different layers. It is nothing but the secondary growth, that is the extrastellar secondary growth. Now this periderm is a three-layered. At the center there is the phellogen, which is the meristematic in the nature, and this layer, that is this phellogen, cuts off new cells from outside and from inner side. Now from outer side it cuts new layer, and that layer is called as the cork or the phloem, and it is irregular in shape, and inside this it cuts off another layer which is called as the cortex. It is also known as the phellogen. It is also known as the phelloderm. So in this extrastellar secondary growth, which called as the cork or the bark, there are the three different layers, that is the phloem, phellogen and phelloderm. Now this is about the extrastellar secondary growth. Now inside this there is the cortex. So this portion is of the cortex, and these are the lip traces. Now from external side of this lip, it is irregular in shape because of scars, permanent scars of the lip. So this is the cortex. They are parenchymatous in the nature, and it stores the food material. Now inside this there is the vascular bundle. Now this is the lip trace, and inside this there is the vascular bundle, and this is the phloem, and this is the xylem, and in between phloem and xylem there is the cambium. So this type of a vascular bundle is called as the conjoined, collateral and open. That is conjoined, collateral and open. That means conjoint. Conjoint means when both xylem and phloem are developed in one and the same vascular bundle, then it is called as the conjoint. When these both xylem and phloem lie side by side, then it is called as the collateral. And if in between this xylem and phloem, if the cambium is present, then it is called as the open. So this type of vascular bundle is called as the conjoint, collateral and open. Now at the center there is the pith, and in the pith there is a mucilage canal are also there. Now it is a parenchymatous in nature, it also stores the food material. Now in between this vascular bundle there is the medullary rays, and the type of stele is ectophoic siphonostele. So this is all the details of the young stem of the cycas. Now about this old stem. Now the structure, external structure is the same. That means that there is the periderm, and periderm again shows the three different types of the layers. They are called as the phloem, phellogen and phelloderm. This is the extrastellar secondary growth. Now inside this there is the cortex. It is again loosely arranged. It is thick walled, and it stores the food material. Inside this there are the lip traces, and from outer side there are also the lip scars. They are found in the periderm. Now at the center there are different number of rings of the xylem and phloem are get formed. So this is the primary ring of the vascular bundle. Now after this, the cambium becomes inactive, but later on it becomes active and it is get formed in between this primary ring of vascular bundle and in between the cortex, and there it first form the cambium ring, and again this cambium ring adds new xylem, that is the secondary xylem, toward the inner side, that means this xylem, and secondary phloem toward the outer side, that means this phloem. In this way a complete ring is get formed, which is called as the secondary growth, that is the intrastellar secondary growth. In this way, this is the first ring, that is the primary ring, this is the second ring, and after this number of rings are get formed and they are seen here. Here these are the two rings, but after this new rings are also get formed, and the number of these ring increases up to the 20 in number. Now at the center there is the pith. Here the cells are loosely arranged, they are thin walled, they are parenchymatous, and they store the food material. So this is about the internal morphology of the tap or normal root system of the cycas. Thank you.